Assalamu alaikum. My name is Hajra Yunus, and today under the topic of molecular biology we will discuss about, or under the subject of molecular biology we will discuss about the very important topic, that is requirements of DNA replication. So before we are going to start the process of replication in prokaryotes and eukaryotes, we will look into the requirements of DNA replication, means what things are required during the process of DNA replication by prokaryotes and eukaryotes. In this lecture, we will cover the topics of requirements of DNA replication, and under the topic of requirements of DNA replication, we will discuss initiator proteins, helicase loader, helicase uh, single-stranded binding protein, primase, and DNA polymerase. So in this slide, we will discuss about DNA replication. So as you know that in the last lecture we discussed about the process of DNA replication. They are different because before that, now we know that the DNA replication mode is semi-conservative in nature, like I have written here. But before that, it was not known that. So there were three modes of DNA replication. One was the conservative, another was the semi-conservative, and third was the dispersive. Now we know that the process of DNA replication is semi-conservative in nature. Semi-conservative, let me tell you again what is semi-conservative DNA replication. In case of semi-conservative DNA replication, out of two strands, where there is a process of replication, so out of two strands, the one strand is a parental strand and the another strand is the newly synthesized strand. Okay, so you can say that the one strand is a template, and opposite to that the complementary strand is formed. So that is the DNA replication, that is a semi-conservative DNA replication. Now you can see that here there are two strands. So this is the one strand and this is the another strand, and then during the process of the DNA replication, two strands get separated and opposite to them the newly synthesized strand is formed. So here, out of two strands, the one strand is the leading strand, another strand is the lagging strand. So what does it mean by leading strand or lagging strand? In case of leading strand, the leading strand is continuous strand and lagging strand is the discontinuous strand. As you can see in this diagram that here the 5' hydroxyl group, 5' phosphate group is present, and here 3' hydroxyl group is present. Okay, so you know that the DNA polymerase works from 5' to 3' direction. So as long as 3' hydroxyl group is available, DNA polymerase can perform its activity or can perform its function. So here the DNA, here the helicase is present. So helicase is performing its function. What is the function of helicase? Helicase is responsible for unwinding of DNA strand. So here the parental strand is from 3' to 5' direction, and here the DNA polymerase is working from 5' to 3' direction, and here 3' hydroxyl group free is available. So here the DNA polymerase can perform its function. So but on the other strand, you can see that there is a it is called the discontinuous strand or lagging strand, because here the parental strand is 5' to 3' direction, and the primer, here the primer was attached, that was 3' to 5' direction, and now here the 5' phosphate group is available. So here at that strand, on that strand, DNA polymerase cannot perform its function in that direction. So here the 3' OH group is available, so there is no space or there is no nucleotide, or opposite to that the DNA polymerase cannot perform its activity. So in that case, here the primer is attached, and now after the attachment of primer, let me remove this. So here the primer is attached, or here 5' phosphate group is present and 3' hydroxyl group is present, and now here 3' OH group is available, so that DNA polymerase can perform its activity. So in that case, the replication will proceed in that direction. So here the DNA replication on this strand is the discontinuous or lagging strand, or here the different fragments are present, or number of fragments is present. That's why it is called the Okazaki fragments also, because the Okazaki fragment name is given based on the scientist who discovered those fragments. And now you can see that here, firstly, the, for example, imagine that, because you need to imagine here, the firstly the unwinding was taken place at this point, and here this region was available, so the primer was attached and the process of DNA replication was carried out. And now after that, now after the completion of the DNA replication, in here the DNA helicase has reached here, and now this space is available for the DNA replication. Now the primase will perform its function, the primer is attached, and now this portion is replicated. So in that case, on the one strand, because this strand is continuous strand, because here there is no hindrance, 3' free hydroxyl group is available. So here on this strand the DNA polymerase works continuously, but here the 3' hydroxyl group is not available all the time. So when the helicase moves forward, so in that case the small area is available for replication. So in that case the primase will perform its activity. Primase is responsible for the formation of primer. So in that case RNA primer is attached, and now 3' hydroxyl group is available. So in that case DNA polymerase starts the process of polymerization. So the next slide is about the requirements of DNA replication. Okay, before we study the process of DNA replication in prokaryotes and eukaryotes, we must know about what are the requirements of DNA replication in prokaryotes and eukaryotes. So here the important things which are required for DNA replication are template, DNTPs like deoxyribose nucleotide triphosphate, initiator proteins, helicase loaders, helicase, single-stranded binding protein, clamp loaders, DNA polymerase, and primase. All these things are required, and in the end, when there is a process of termination, at that time few proteins are required, like in case of prokaryotes tertus proteins are required, and in case of eukaryotes telomerase enzyme is required. Okay, so now we will discuss each and every step. Now we will discuss each and every point step by step, and each and every requirement step by step. So this is the template and DNTPs. Okay, as you can see in the first diagram, in this diagram here, the template means out of 2 strands. So here, before the process of DNA replication, let me change color. Okay, so the DNA was like this. Okay, so the template is available. So after the process of replication, when before that only one copy of the DNA was present, so after the process of DNA replication 2 copies were present. So this is the one copy and this is the another copy. So for that the template is required. So here you can see, so this DNA, one strand of DNA acts as a template for one strand, and this strand of the DNA acts as a template for another strand. So here you can say, after the activity of the DNA of the helicase, here the activity of the helicase, now the strand has been unwinded. So out of 2 strands, this strand is the template, will act as a template, and this another strand will also act as a template for the leading strand, and this strand will act as a template for lagging strand. Okay, and next is the DNTPs. What are the DNTPs? DNTPs are the deoxyribose nucleotide triphosphate. So in that case, you can see that during the process of DNA replication, there are 4 types of DNTPs. Number 1 is the adenine, that is the double ring structure, and guanine is also the double ring structure. Cytosine is the single ring structure and thymine is also the single ring structure. So these four DNTPs are used during the process of DNA replication. Okay. Next is the ORI sites and initiator proteins. So firstly, uh, before the start of the DNA replication, we should note about that what are the ORI site. So what is the ORI site? Is the origin of replication, and in case of prokaryotes, the origin of replication is called the ORIC, whereas in case of eukaryotes it is called the ARS. Okay. So in case of prokaryotes and eukaryotes, there are different names for ORI site in case of prokaryotes and eukaryotes also. And you can see here, this is the replicator. It shows that this is the origin of replication. And origin of replication in case of prokaryotes is 200 to 250 base pair long, and in case of eukaryote it is 100 base pair long. So why in prokaryotes the origin of replication ORI site is 200 or 250 base pair long, that is more base pair. In case of eukaryotes the base pairs are less. The answer is that it depends on the nature of DNA and size of DNA also. Like in case of prokaryotes, the size of DNA is less, that's why the origin of replication is just one. So in case of prokaryotes there is only one DNA or origin of replication, one ORI. Okay, whereas in case of eukaryote there are multiple DNORI site. So in case of prokaryotes, the ORI site is 200 to 250 base pair long, and in case of eukaryotes ORI site is 100 base pair long, and in case of eukaryotes ORI sites are many in number, and in case of prokaryotes the ORI site is only one in number. So this is the difference between prokaryotes and eukaryotes, and that is a very important difference. So another thing is that, so here the ORI site is available. So firstly, this ORI site is recognized by the initiator protein. So this is the initiator protein, as you can see in this diagram. So this is the initiator protein. So in case of prokaryotes, the initiator protein has different name, and the structure is also different. And in case of eukaryotes, the initiator protein has different name and different structure. So now we will look into that, what is the name of initiator protein in case of prokaryotes and what is the name of initiator protein in case of eukaryotes. So as you can see that here, in case of prokaryotes the initiator protein is called the DNA. Okay, and in case of eukaryotes the initiator protein is the ORC16. So it is the abbreviation of origin of replication complex 16. So there is one important thing for both of them is that in case of initiator protein, whether we talk about the prokaryotes and eukaryotes, so the both initiator proteins are of 6 subunits. Okay, so these are of 6 subunits. So now you can see in this diagram here the origin of replication is present, and this ORI site is recognized by the initiator, and after the recognition by the initiator, so initiator is responsible for melting the hydrogen bond between the hydrogen bond present at the ORI site, because at ORI site 80 ORI site is 80 rich. So ORI site is, if ORI site is 80 rich, in that case the melting temperature is low, so it is easy for initiation protein or initiator protein to break the hydrogen bond between the two strands. So now here you can say here the single strand is available. So helicase cannot be loaded until there is a single strand. Helicase cannot be loaded on the double strand. If there is a single strand, and then helicase can be loaded. So all these preparations are done for the loading of helicase, because if there is a single strand then helicase can be loaded. If there is no single strand then helicase cannot be loaded. So in that case, the firstly, let me repeat one time again. Firstly, there is a ORI site. ORI site is recognized by the initiator protein. In case of prokaryotes, the initiator protein is DNA, and in case of eukaryotes, the initiator protein is ORC16. So after the recognition, so the ORI site has high AT content, and because of high AT content the melting temperature is low. Because of the low melting temperature, it is easy for the initiator protein to break the hydrogen bond between the two strands to make it single strand, and now there will be the functioning of the helicase loading. So helicase loading will be done after that. Next is the helicase loader and helicase loading. So for the loading of helicase, helicase loader is required, and if there is no helicase loader, there is no loading of helicase. So as you can see in this diagram, in case of prokaryotes, the helicase loader is called the DNAC, whereas in case of eukaryotes, helicase loader are of two types. Number one is the CDT1 and other one is the CDC6. There are two helicase loaders in case of eukaryotes. In case of prokaryotes, there is only one helicase loader. Next is the, as you can see in this diagram, but let me tell you one thing before. In case of next is the helicase. In case of prokaryotes, the helicase is the DNAB, and the eukaryotes, the helicase is MCM27. So you have to remember all these things or all these names. So after 2 to 3 times when you memorize them, it will be easy for you to it will be easy for you. Okay, so in case of next first one, helicase loader is required for the loading of helicase. As long as helicase loader is available, helicase can be loaded. If there is no helicase loader, so the helicase cannot be loaded. So these both things are very important. So this diagram is for the prokaryotes and eukaryotes. As you can see, in case of prokaryotes, the helicase loader is the DNAC. So as you can see in this diagram, there is a helicase loader, that is the DNAC, and this one is the helicase. So before the loading of helicase loader will attach, and the helicase loader can hold the helicase. So in this diagram you can see that there is a, let me remove this color. Okay, as you can see in this diagram, firstly there is a loading of helicase loader, and after that there is a loading of helicase, because helicase loader provides space for the loading of helicase. So now you can see that in this diagram, the loader is the helicase is the MCM2, and in that case, in case of the eukaryotes, there are two helicase loader. Number one is the CDC6, another one is the CDT1. Okay, so firstly, out of these two helicase loader, we will study how the helicase loaders are loaded in case of eukaryotes and prokaryotes. But let me tell you one thing here. The firstly, out of these two helicase loader, firstly CDC6 is loaded, or CDC6 loads, and after that CDT1 carries the actual helicase along with it and loads on the DNA strand. So we will study the detailed process, how the helicase loaders work, and how the helicase enzyme is loaded on the DNA strand with the help of the helicase loaders. So this slide shows that helicase loaders in case of the prokaryote is only one, that is DNAC, and in case of eukaryote there are two, that is CDT1 and CDC6, whereas in case of prokaryotes the helicase is called DNAB, and in case of eukaryotes is called the MCM27. And the one thing is that in case of the before the loading of helicase enzyme, helicase loader is loaded, and after that the helicase loader can hold the helicase enzyme. And in case of prokaryotes there is only one, and in case of eukaryotes there are two loaders. So out of two loaders, firstly the one loader is loaded, and another loader, that is the CDT, that carries the helicase on the DNA strand, and after loading the DNA strand, both the loaders, that is CDC6 and CDT1, they will leave the DNA strand. And when we discuss the process of DNA replication in the eukaryotes, we'll discuss about how CDC6 and CDT1 will leave the DNA strand. So their process is a little bit different. They will leave after using the ATP. So we will discuss how CDC6 and CDT1 will leave the DNA strand. Next is the polarity of helicase. This is very important slide about the polarity of helicase. So as you can see in this diagram, in case of so this is about the prokaryotes and this side is about the eukaryotes. So as you can see in this diagram, the polarity of the helicase is from 5' to 3' direction, and in case of eukaryotes it is from 3' to 5' direction. So what does it mean by 3' to 5' direction and 5' to 3' direction? So the thing is that in case of prokaryotes, the helicase is loaded on the lagging strand. Okay, so lagging strand. So whereas in case of eukaryotes, the helicase is loaded on the leading strand. Okay, so this is the polarity of the helicase. In case of prokaryotes, the helicase is loaded in 5' to 3' direction, as you can see here, 5' to 3' direction, and here, or you can say the helicase is loaded on the lagging strand, whereas in case of eukaryotes, the helicase is loaded on the polarity of helicase in case of the eukaryotes is the 3' to 5 direction, or the helicase is loaded on the leading strand. So these two things are very important when anyone asks you about when there is a question regarding the polarity of the helicase, then you should know about the polarity of helicase in prokaryotes and eukaryotes, because polarity of helicase in prokaryotes and eukaryotes are different. In case of prokaryotes, the polarity of the helicase is from 5' to 3' direction, whereas in case of eukaryotes it's 3' to 5' direction. So next step is the single stranded binding protein. So single stranded binding protein, what is the single stranded binding protein? You can see in this step, like this is the DNA strand, and here after the activity of the helicase, here the two strands get separated, but you know, these two strands may try to come closer to one another and may try to form the hydrogen bonding. So to prevent the hydrogen bonding and to prevent these two strands to again form the hydrogen bonding, so here you can see that here these something green balls are present. So these green balls are actually the single stranded binding protein, and these single stranded binding proteins are responsible to prevent the rewinding of DNA strand. Okay, if the single stranding binding proteins were not present, in that case the DNA strands may join together or may rewind. So in that case the process of DNA replication may be affected, but because of the presence of single stranding binding proteins, these single stranding binding proteins are responsible for the or these single stranding binding proteins do not rewind the DNA strand. So because of that the DNA strands get separated or remain separated. So here in case of prokaryotes, single stranded binding protein is called the SSB, whereas in case of eukaryotes it is called the RPA. Okay, so there is one more thing that is very important, that is the in case of single stranded binding protein like this one, single stranded binding protein has two important properties. One is that the electrostatic interaction is present with phosphate backbone. So the electrostatic interaction is weak interaction, that can be the van der Waals interaction or hydrophobic interaction. Suppose it were a covalent interaction, in that case the after the attachment of the single stranding binding protein or SSB or RPA, if they would not get separated, so in that case the process of DNA replication would be affected. So but now the weak interaction is present, that is the hydrophobic interaction or the van der Waals interaction is present. So in that case the SSB or single stranding binding proteins get separated and the process of the DNA replication continues. And another important point is that they are not sequence specific, means it does not matter what kind of nucleotide is present. Their function is just after the activity of the helicase enzyme, when the helicase enzyme unwinds the DNA strand, after that the single stranding binding proteins go and bind to the DNA strand or attach to the DNA strand to make sure that these two strands again not rewind. So it doesn't matter what kind of nucleotide is present, so their function is just to prevent the rebinding of DNA strand. So these two things are very important, that is the electrostatic interaction with phosphate backbone, and the electrostatic interaction is weak. Another is that the not sequence specific. Next is the DNA polymerase. So in case of as you can see in this diagram, like in case of prokaryotes, 5 polymerases are present, and here they have written that the 5 polymerases are present, but actually in eukaryotes these 5 polymerases are important, but there are many polymerases, like more than 12 or 15 polymerases are present. Like if I call the name, that would be theta and sigma and neta, eta, zeta, and so on. There are many polymerases are present, but these 5 polymerases in case of eukaryotes, and in case of prokaryotes 5 polymerases are present, not more than that, but these 5 polymerases have their important function. Let's study about these polymerases one by one. Like in case of polymerase 1 is involved in synthesis, proofreading, repair, and removal of DNA primer. Okay, so the first step is the synthesis, proofreading, repair, and removal of DNA primer. So before the process of polymerization, because DNA polymerase cannot perform or cannot initiate the process of polymerization, so the DNA primer or RNA primer is required. So in vivo system the primer is the RNA primer, but in vitro system the primer is the DNA primer. So DNA polymerase the process of DNA replication or DNA polymerase can work with either the DNA primer or RNA primer. Okay, so it doesn't matter what kind of primer is present, it can be RNA primer or it can be DNA primer. Okay, so the primer is synthesized by polymerase 1 in case of prokaryotes, and after the process of the or after the completion of the DNA replication, primer is removed also by the polymerase 1. So these two things are very important. You need to know that in case of prokaryotes, the primer is synthesized by polymerase 1, and after the completion of DNA replication, primer is removed by polymerase 1. And if there is a mistake, this mistake can be proofreaded by DNA polymerase 1 also. Okay, and it has repair property also. Next is the polymerase 2. Polymerase 2 is also a repair enzyme. So actual for the during the process of DNA replication, actual DNA polymerase is the polymerase 3. So it has written that the polymerase 3 is main polymerizing enzyme, and now the polymerase 4 and 5 are the repair enzyme under the unusual condition. So what does it mean by unusual condition? So when the condition is such like that, when there is the process of the DNA replication is not going on, in that case the polymerase 4 and polymerase 5 will take over the process of polymerization. But there is a one difference between polymerase 3 and polymerase 4 and 5. So the difference is that, and that is a very important difference, the difference is that if there is a mistake, so polymerase 3 goes back and repairs the mistake, but in case of polymerase 4 and 5 they can perform the process of polymerization, but if there is a mistake they will not go back and will not repair the mistake. So there is a very important difference between polymerase 3, polymerase 4 and polymerase 5. Okay, I hope everything has is you're understanding each and every point. Okay, I'm trying to make every point clear to you, that it will be easy for you when you go through the slide later. It will be easy for you, or go through the lecture, it will be easy for you to understand that what is the difference between polymerase DNA polymerases in case of the prokaryotes and the DNA polymerases in case of the eukaryotes. Okay, so in case of eukaryotes, the alpha polymerase a polymerizing enzyme, and it is responsible for primer formation. Okay, it is responsible for primer formation. Polymerase beta is a repair enzyme. Polymerase gamma is the mitochondrial DNA synthesis. It takes part during the synthesis of DNA of the mitochondria, and that is the epsilon, and that is the delta and epsilon. Sorry, delta and epsilon. These are the main polymerizing enzyme. Okay, here it is written that the function is unknown, but actually they are the main polymerizing enzyme. They both are the main polymerizing enzyme. Okay, so I hope each and everything is clear to you. So there is a difference between the prokaryotes and the eukaryotes, but when we talk about the actual or the main polymerizing enzyme, so in case of prokaryotes, the main or actual polymerizing enzyme is the DNA polymerase 3, whereas in case of the eukaryotes, the main or actual polymerizing enzymes are the delta and epsilon. Other polymerase DNA polymerases have their own function. Like this one is also very important. Like if there is no alpha polymerase, so in that case there will be the no RNA primer. If there is no primer, so the process of the DNA polymerization will not carry out. But the thing is that, so I'm talking about in that content that which DNA polymerase is responsible for the process of polymerization. So in case of prokaryote, DNA polymerase 3 is responsible for the process of polymerization, and in case of eukaryotes, DNA polymerase delta and epsilon are responsible for the process of DNA polymerization. Okay, so now there is one more thing. This diagram illustrates the difference between the processive and non-processive DNA polymerase. Okay, like in case of prokaryotes, DNA polymerase 1, and in case of eukaryotes, DNA polymerase alpha, they both are the non-processive DNA polymerase. And the actual DNA polymerases which are responsible for the polymerization of the DNA replication or the DNA strand, they are the processive polymerases. So what is meant by, or what is the difference between the non-processive and the processive DNA polymerase? So here you can see in this diagram, so this side shows the non-processive nature of DNA polymerase, and this side shows the processive nature of DNA polymerase. In case of non-processive DNA polymerase does not have the clamp. Okay, there is no clamp. If there is no clamp, so in that case, after adding few nucleotides, the DNA polymerase will fall down and process of DNA polymerase or DNA replication will be halted. Okay, so in the next diagram we will look into the structure of the DNA polymerase and the clamps, on how the clamp can hold the DNA polymerase. In that case, the DNA polymerase 3 or DNA polymerase delta or epsilon will not fall down. So whereas processive, in case of the processive DNA polymerase, as you can see here, in case of the processive DNA polymerase, there is a clamp is available. So that what is the function of the clamp? Clamp can hold the template strand when something holds the template strand in that case, and the DNA polymerase attaches to the clamp. In that case, the DNA polymerase 3 or DNA polymerase delta or epsilon will not fall down. So in case of the non-processive, there is no clamp, and whereas in case of the processive, there is a clamp. So as you can see in this diagram, so you can imagine that if there is no clamp, so after adding few nucleotides, here you can see that the DNA polymerase will release or will fall down, and the process of DNA replication is affected. Whereas in that case, here the DNA polymerase is attached to the clamp and the clamp is connected on the template strand. So in that case, the process of DNA replication will go smoothly, and after completing the DNA strand, the DNA polymerase releases itself. Okay, so this is very important slide. What is the difference between the processive and the non-processive DNA polymerase? So this is the structure. So this is the clamp loader, and the clamp loader is very important for the loading of the DNA polymerase. As you can see in this slide, there is a clamp loader, and the clamp loader is attached to the sliding clamp. As you can see, this sliding clamp has this sliding clamp will attach to the template stand, and here you can see this is the polymerase. So this is the diagram of the prokaryotic. So this is the polymerase 3 core, and here the two are, this is the flexible linkers, or actually this diagram shows only two linkers, but actually there is a sliding clamp, and the sliding clamp is attached to the clamp loader, and the clamp loader has 3 arms. So let me make one more arm. So here you can see here the one more arm is present. Actually in this diagram they showed only 2 arm, but actually 3 arms are present, or here the one more DNA polymerase 3 is available. So these arms are, out of 3 arms, 2 arms are flexible. Okay, so here this one arm and this arm, these 2 arms are flexible, whereas the one arm is not flexible. So why 2 arms are flexible and the one arm is not flexible? The reason is that here these 2 flexible arms are responsible for the polymerization on the lagging strand, whereas this arm here, the arm is not flexible, and this arm is responsible for the polymerization of the DNA strand on the leading strand. So the leading strand is continuous, so because of the continuity the flexibility is not required, whereas on the lagging strand, so these two polymerases, DNA2 polymerases 3 work together, so in that case there is a flexibility, so they can change their position. So after completing their work on one strand, they will move to the another strand, and then they are moved to the another part. So in that case, if there is a flexibility, so after completing work on small part, they can move to another part to complete the process of polymerization and the DNA replication on that part. So that's why these two strands are flexible. So this is the structure of the clamp loader. Like in case of the clamp loader, the sliding clamp is attached. Thus this sliding clamp and the clamp loader, they can hold the template stand. In that case, the DNA polymerase 3 or the DNA polymerase delta or epsilon in case of the eukaryotes will not fall down, and the process of DNA replication will go smoothly. There will be no problem. So here let me repeat this slide again. Here sliding clamp is present and the clamp loader is attached to sliding clamp. Here the 3 arms are present. Out of 3 arms, 2 arms are flexible, 1 arm is not flexible, because flexibility is not required for the polymerization of leading strand, whereas flexibility is required for the polymerization of lagging strand, because the leading strand synthesis is continuous, whereas the lagging strand synthesis is discontinuous. So after completing its work on the one strand, the DNA polymerase and this arm can move to another position to complete the process of DNA replication on that portion. So this is the structure.